Okay, selective media. This is what you want because the substances favor or inhibit growth of particular or very specific microorganisms. So here's an example of selective media. Here's bacterial colonies at 7.3. Here's fungal colonies at 5.6. This is the same media. You can see there are different things that grow at 7.3. So it's selective. Enrichment culture, selective media used to increase small numbers of microbes to observable levels. Sometimes you can do a cold enrichment. This is enrich a culture with cold tolerant species. If it grows better in the cold, then that's a way to think about the selective media. Blood agar is used as a differential media. Carbohydrate utilization tubes, Durham tubes, we will do these, no fermentation. You will do a couple of these a lot. Here's some MacConkey, you will use that. This is the differentiation of enteric bacteria based on ability to ferment lactose. So you will order a bunch of MacConkey agar. We will use it. It looks like this. MacConkey agar, here's your Staphylococcus, here's your E. coli on regular nutrient agar. Here's some MacConkey agar, Staphylococcus aureus, no growth, E. coli, fun. Here's E. coli again on the MacConkey agar. Here's Salmonella. It turns a really funny color. So this MacConkey is one of the most prominent differential medias, selective medias that you will use in class. Now, we do not have one of these anaerobic culture systems, so we're not going to do it like this. We can't really create an anaerobic media like this. So if we're not going to give you any anaerobic bacteria because we wouldn't have a way to keep them cultured. Transport media, this is used by health personnel to ensure specimens are not contaminated. There's a bunch of different techniques here. We are really just going to use the one that we have been talking about in class. We don't have to use deep freeze it to store that for years, stores for decades. Refrigeration, we're going to store your samples in the refrigerator. It will stop the growth and it'll store them for the 2 or 3 weeks that you need them stored. Why do clinical lab scientists keep many different kinds of culture media on hand? You can pause it, talk to your neighbor. Basically because a large variety are capable of colonizing the body, and they range from opportunistic to fastidious. So different selective media are needed to isolate and grow large numbers of colonies. Most microorganisms reproduce by binary fission. They just kind of like pull in half, like it's one, and then they just become two, and now there's a whole bunch of them. It involves basically 4 steps. I don't know if that's going to play. Let's see. No, it's not going to play. So we're just going to look at it like this. Essentially, it's just, remember a lot of these don't have a specific nucleus, so they're just going to fuse and make a bunch of colonies from themselves. Just asexual reproduction. Your garden variety asexual reproduction. Here's another picture of it. This one won't play either. We are going to talk about generation time. Time required for a bacterial cell to grow and divide, dependent on chemical and physical conditions. Usually time and temperature. So here's time in hours, number of cells. This obviously has 2 different growth periods. This, I feel like this is on a multiple choice quiz or something. So this is the lag phase. So there's a minute, it's not going to start growing. You can't come back 30 minutes later and your bacteria is growing. Probably. It has this exponential phase, right? This is usually 4 to 8 hours. This is why we don't. So then you have a stationary phase, which is fine. It just kind of stagnates and stays, and then it starts to die. So we like to take your bacteria out of the incubators at about, if you do it on Tuesday, we take it out on Thursday. If you do it on Thursday, going all the way to the next Tuesday, you're going to start to see some decline, and it's going to be a big old mess. So this is the lag phase. It's just kind of ramping up, then exponential, then it sort of stays the same. It's not dead here. It's just not exponentially growing, and then it declines. It's not, again, it's not dead. It's just declining.
Bacteria is defined as an increase in the number of bacterial cells within a population. Growth trends within a bacterial population can be illustrated using a bacterial growth curve, such as this one. Growth occurs in 4 phases: lag, log, stationary, and death. Let's look at each phase of the curve to see what's happening. When dormant bacteria are transferred to a fresh medium with plenty of nutrients, they spend some time switching their metabolic machinery from the dormant state to an actively growing state. During the lag phase, the number of new bacterial cells is equivalent to the number of dying cells, resulting in a horizontal plot on the growth curve. The lag phase can last from less than an hour to days, depending on the species of bacteria. During the log phase or exponential growth phase, the bacteria are actively undergoing binary fission.
Right?
During the log phase, the bacteria double their numbers every generation period. The generation period can range from 20 minutes to days, depending on the species. As long as there are plenty of nutrients and little waste buildup, the bacteria will continue to grow exponentially. It's convenient to represent the growth of the bacteria on a logarithmic graph because the numbers of bacteria increase very rapidly with each subsequent generation. When the log of the number of cells is plotted versus the generation, the graph is linear. After the bacteria have grown for a while and they're starting to deplete the nutrients in the growth medium, some of the cells begin to die. The stationary phase is reached when the number of new cells equals the number of cells that are dying.
The number of new
After the
equal
bacteria
the number
have
of dead.
depleted most of their nutrients and their waste buildup is getting too great, the bacteria begin to die in larger numbers than are made. If the death phase is carried on for a long time, most of the cells will die. If a cell is to survive, it must switch its metabolism back to a dormant state because there are not enough nutrients available for any more growth.
Endospores. Okay, now why do we care about these growth phases? Because we want to know how bad the microbiology population is. Like, how severe is the infection? How effective is your food preservation? Are you actually still growing bacteria in the refrigerator? The measure of the contamination of the water supplies. So knowing where we are and how many microbes, kind of important. Mostly direct methods not requiring incubation, just your plain old microscope counts. So essentially, we do have these counter cells for estimating microbial numbers. We probably will not get to this lab, but essentially you're just counting them and then doing an exponential growth chart to see how many there are. Obviously, there are electronic ways of doing this. A Coulter counter counts every cell. There's, really fancy ways of doing it. We are obviously not going to do it that way. If we did it at all, we would do a serial dilution and look at the most probable number, just extrapolating, right? So we'd put one culture and then extract until we got 0 colonies, and then we would just estimate the number in here. Does that make sense? Right, if this is a 1 to 10,000 ratio and we've got 1, 2, 3, 4, 5, 6, then we have 60,000. Yeah, everybody good with that? A Coulter counter just catches it in here, and again, probable number for testing, and essentially measuring microbial growth can also be done in a turbidity situation where you look at remember when we did the broth and you kind of look to see how foggy it was? That's turbidity, and it can also estimate population. You can put it in a turbidity counter and it will give you an estimate of population. Students transfer some gunk from a 2-week-old bacteria into new media. Why shouldn't they be surprised when the death phase sample grows? Because it's not dead. It just means the rate of cell death exceeds the rate of new cells. Not that all the cells are dead. Many bacteria become metabolically inactive. They become endospores. So just because you leave it in there for too long doesn't mean it's completely dead. Okay, that's all of them. Bye.